Zara coming to talk to us from the Plymouth Marine Lab and also the University of Essex. And she'll be talking to us today on the effects of microplastics on zooplankton. Just to remind everyone, try to keep your mic and video off for the seminar. And then any questions, you can either post them in the meeting chat or you can raise your hand, and questions will be answered at the end. And if you've raised your hand, I'll call on you to unmute yourself and talk to ask your question. Okay, so over to Zara.
Thank you, Claire. Hello, everybody. My name is Zara Botterell, and I am just going to hopefully share this with you. So I hope that you can all see my presentation right now. Yeah, if not. So my name is Zara Botterell. I am a third-year PhD student based at Plymouth Marine Laboratory. And today I'm going to be talking through kind of like the first half of my PhD. So looking at the bioavailability of microplastics to marine zooplankton. And in particular, I'm going to focus on the effect of shape and infochemicals. So I'm sure you're all used to seeing pictures very similar to these. Plastic is a really wonderful product because it can be used for anything. It's revolutionized technology and healthcare, but the same properties that make it so useful in our everyday life mean that it also can last a really, really long time, especially when it's thrown away. So this picture up here is actually taken in the Caribbean, and this is all big microplastics. Macro pieces are bigger than 5 millimeters that just basically form these large mats, and these can cause problems for many different species that can ingest this, or like this poor turtle here that can become entangled with it. And this picture here of this seahorse, which is using this cotton bud instead of a piece of seagrass, I think was in the National Geographic, really highlighted the problem that these big pieces of plastics can have once they enter the marine environment. But it's not just the big pieces of plastic that we're interested in. We're also interested in small pieces of plastic termed microplastics, which are between the size range of 1 micron and 5 millimeters. So where do these small microplastics come from? Well, there's many different sources. I've put a few up on the screen here. This top left-hand one here are pre-production resin pellets, plastic resin pellets, also termed mermaid's tears. If you've been on any beaches, particularly in the southwest of the UK, you may have found them. I found them pretty much on any beach I've been on. These are the raw materials that they basically melt down to make your plastic bottles, plastic bags, anything. And they're often transported around the UK, well, around the world, especially around the UK, in container ships. And because of stormy weather, they can be spilt overboard. And because they're plastics there and small, they easily float and wash upon our beaches. Another source is, and one that's probably going to start decreasing since the recent ban, is microplastics that are present in exfoliants, makeup and cosmetics that recently they've been banned, but because they were so small, they would go straight down our drains into our wastewater treatments. And again, because they're very small, they go through the mesh that's meant to catch things and head straight into our waterways and then straight out to sea. This middle one here is when we wash our synthetic clothing, so nylon and polyester clothing items, when we wash them, they get a lot of friction in the washing machine and they cause them to shed fibres. Again, the same problem with the cosmetics is because these fibres are so small, they don't get caught by mesh and they end up into our waterways and marine environment. This one down here is the wear of tyres. So as we're driving around, our tyres become worn down and this sheds into our ditches and drainage systems that again enter the waterways and end up commonly found in our oceans. And finally, this last picture has two sources of microplastics, one of which is obvious. It's this large piece of plastics here that through UV degradation, physical abrasion, will break down into smaller and smaller pieces of plastic. And the last one is actually looking at this ship here, which has anti-fouling paint, which contains paint particles. And as we all know, boats become chipped and weathered, and this is also released into the marine environment. And we're particularly interested in these small plastics because the smaller they are, the greater number of organisms that they can interact with, which includes those of zooplankton, which are a very important group of marine organisms at the base of the marine food web. So these are a very important food source for many other species, including fish, whales, seabirds, and they're also very important for nutrient cycling. So they take this fixed carbon, they eat phytoplankton, which fixes carbon, the surface of the ocean, and through their vertical migrations and through producing fecal pellets, they sequester this carbon in deeper water. So they're essential for food and for nutrient cycling. So what do we already know about zooplankton and microplastics? What we do know is that microplastics are readily ingested by zooplankton. These images here are from Cole et al. 2013, and you can see that many different species, so these are copepods, these ones here are crabs, and also bivalves, so mussel larvae, have all shown to ingest plastics, which is this green, yellow, fluorescent inside all the organisms, and it's also found in their fecal pellets, which is the ones down here. Further research has shown that microplastic ingestion has led to decreased feeding, reproduction, and survival. However, there's currently very little research investigating the mechanisms behind plastic ingestion, i.e. its bioavailability, why the zooplankton are eating it, what factors are making this seem like it's a food source or being conviewed for a food source. So for the first part of my PhD, I conducted a literature review where I looked at the bioavailability and effects of microplastics on marine zooplankton. So the first part of it, I assessed all the recent literature, looking at which studies had assessed what factors and using which microplastics, which species. And I found that negative effects were reported in 45% of studies, 14% reported none. The majority of these studies occurred within the laboratory, and microplastic ingestion was recorded in 32. Oh, what are you doing? Microplastic ingestion was recorded in 32 species of zooplankton. 23 are holoplanktonic. So this means that these are species of zooplankton such as copepods, which spend the entirety of their life cycle in the zooplankton. 9 were meroplanktonic. So these are species such as crabs and fish larvae, which have a life stage within the zooplankton. For the next part of my literature review, I then assessed the factors which could affect the bioavailability of microplastics to zooplankton. And there's many different ones. So there's the obvious ones, which you can probably kind of guess, which would be the abundance, the size, and also the selectivity of the species themselves. But maybe some different ones also, such as shape, the ability for microplastics to aggregate, colour, and also age, so this is the length of time plastics are in the water. They can develop a biofilm. They can become cracked, they can be weathered. And I'm actually going to come back to this factor in a few minutes' time. The presence of a biofilm can also affect the density. So not only do you have the density of the plastic polymer itself, but the fact that if it becomes aged, it can become heavier. So looking at all these previous studies and then looking at these factors which can affect the bioavailability, it became very clear that the microplastics used in laboratory studies were pristine, a single polymer type, and of uniform size, shape, and colour. And I don't know about any of you, but if you've walked along any of the beaches nearby and you've seen microplastics, you'll know that they are not like this at all. They vary completely, and they're all very different. So in this literature review, I also made 6 recommendations for the future of zooplankton and microplastics research. Number 1 was more field studies. Field studies are quite tricky, and there hasn't been that many globally. especially in comparison to lab studies. It's difficulty in sampling techniques, managing contamination, and things like that. Number 2 is to use microplastics in the laboratory that are more representative of those in the marine environment. A lot of previous research has, again, only used certain types of microplastics. And whilst that research is really important because it highlighted the problem, it is now really important that we move forward using more environmentally relevant microplastics. Number 3 is to include a wider range of zooplankton species in life stages. A lot of these, as I previously said, studies were conducted on holoplanktonic species, so i.e. those that spend the entirety of their life cycle in the zooplankton, and they were mostly done on adults. It's really important that we maybe include more meroplanktonic species that are important constituents of our finfish and shellfish stocks, and also looking at larval developmental stages, because these are arguably one of the most vulnerable life stages, and any effects here could have lasting effects for the species' life. Number 4 is to investigate bioaccumulation. This would be bioaccumulation of the plastics themselves, how likely they are to retain it, how likely they are to pass this on to further trophic levels. Number 5, to investigate chemicals associated with plastics. So this is a tricky one because a lot of the plastics in the marine environment will absorb chemicals from the marine environment, but each plastic polymer has its own chemical signature as well. So not only do you have chemicals in the marine environment coming to the plastics, but the plastics themselves, and it can be difficult to differentiate where those chemicals have necessarily come from. And lastly, number 6, which is microplastic risk assessment on zooplankton and the ecosystem. A lot of the previous recommendations I've made are kind of required before we can really look into this, but it's actually looking at how much of a problem are microplastics to zooplankton. For example, do they get retained for days, or do they just pass straight through them and end up in the fecal pellets? And what effects this could have on the zooplankton, and what this means not just for the individual, but for the species level and the population, and therefore the risk to the species and the ecosystem. And this could have important implications for policy as well. So for my next chapter of my PhD, I decided to focus on doing some experiments and using microplastics that are more representative of those that are found in the marine environment. So for the next chapter, I asked 2 questions. Number 1 was, does the shape of the microplastic affect ingestion rates by zooplankton? So I chose beads, fibres and fragments. Beads because they are the ones that are commonly used in the laboratory, and fibres and fragments because this is what we commonly find in the marine environment. My second question, I really wanted to look into the effect of age on microplastics. And you remember before I was talking about biofilms, and biofilms have kind of two arms to it. There's the formation and the visual and the visual effects of the biofilm and how it affects and distorts the density of the plastics. But then there is also not just the visual one, but how the biofilm changes how the plastic smells. So biofilms are formed by microbial and phytoplankton interactions on the surface of the plankton, the surface of the plastics. And this can then alter, it really wants to change my colour scheme. This can then alter how this smells. So if you've ever been for a walk on the beach, you'll have smelt this classic smell of the sea. And this is actually due to DMS. So this here is a picture from near my hometown. Maybe there's lots of seaweed on the beach and you've got that classic salty sea air. And that's due to an infochemical, which is called DMS. The infochemical DMSP is produced by phytoplankton, which is a food of copepods and other zooplankton. And when these phytoplankton die or lies, bacteria in the water can convert this DMSP into DMS. Previous research has shown that DMS stimulates grazing in copepods. And I was interested to see whether this infochemical, which is derived from phytoplankton, could also increase and affect the ingestion rates of microplastics by zooplankton. This is my overall methodology kind of design. The species I used was Calanus helgolanthus, which is a common copepod that's found commonly in the North Atlantic, Homerus gammarus, which is European lobster larvae, and Acacia tonsa, which is also another copepod species, which is found worldwide. I chose 3 different species because I wanted to look at different species and their different selectivity, but also each of these different species has different feeding mechanisms. So, for example, Calanus are suspension feeders, Homerus gammarus are ambush feeders, and Acacia tonsa do a little bit of both. I tried to use microplastics that were all the same size, and they were all the same color, they were all yellow. However, my beads were polystyrene and my fibers and fragments were nylon. And this is just because currently on the market, I couldn't find any nylon beads. My infochemicals, as previously said, were DMSP and DMS. And I ran a classic feeding experiment, which was 6 hours in the dark on the plankton wheel for the copepods and 3 hours for lobster larvae. And this is because lobster larvae are cannibalistic, so they had to be run slightly different sized bottles and therefore a smaller time. No algae was included in my treatments. This is because obviously they could produce DMSP and DMS, which could then contaminate them. And then I used FlowCam to generate a concentration of microplastics from which I was able to generate ingestion rate. This is an overview of how I added my bottles and the different treatments that I had. So I had virgin, microplastics and DMSP, microplastics and DMS. And what I did was I pre-infused my infochemicals with my plastics for roughly 3 days before. And then I added them to the bottles. All microplastics I aimed for roughly 80 particles per milliliter. My plastics were pre-incubated with 20 nanomolar of DMSP and 5 nanomolar of DMS. These concentrations are what you typically find at the sea surface during the summer months in the North Atlantic, so they're representative of what we'd find in the marine environment. The concentration of microplastics is probably an order of magnitude higher than what we'd find in the marine environment. This is at the lower scale of plastics that are used in experiment studies at the moment, but it's getting that balance between using things which you're going to be able to see an effect and using things which are not absurd in their concentration levels. So going forward now, looking at the results, we'll first look at those from my microplastic shape, so addressing my first question. We show that in 3 different species, so the Calanus, the lobster larvae, and Acacia, each species preferentially ingested one shape more than the other. So Calanus had more fragments, lobsters more beads, and Acacia more fibers. Why this is, I'm not particularly sure. More behavioural studies will probably be needed, but it's likely to be factors such as the feeding mechanisms of the species, also the similarity to maybe preferential prey items for each of the species, and it could just be ease of handling and ingestion that could maybe affect this. Similarly, the Acacia was the smallest species used, and the fibers had a diameter of 10 microns, so maybe that made things easier to eat. I'm not entirely sure. Now, going forward and there we go. Looking at my infochemical results. This is the set of results for my Calanus. We show on all 3 microplastic shapes that DMS infusion significantly increased ingestion rates across all microplastic shapes. You also notice on these graphs here that I've got I call them my secondary controls. So we have our virgin here, and then I quickly realized that after completing my lobster experiment that I really needed to include a secondary control that didn't infuse my DMSP and DMS, so that we could look at whether it's the presence of the infochemicals in the water versus the presence of the infochemicals maybe on the plastics. So that's what you're seeing there. And crucially, there was no significant difference between the virgin treatments and the control injection treatments, but there was a significant difference between the injection and infusion treatments. Now looking at my lobster experimental results for infochemicals, we also found that DMS infusion significantly increased ingestion rates for fibres and fragments, but neither infochemicals had an effect on the ingestion rate of beads. And this is quite an interesting finding. It's possible that shape is more bioavailable than the infochemicals in this species, and that they preferentially ingested beads was their favourite thing to eat, and they may just prefer the shape of beads. Lobsters also have much better eyesight than the other two species that I used, and it's possible that that may play a role as well. And then my final species, my Acacia, again here we show that DMS infusion significantly increased ingestion rates of fibres and fragments. Again, there was no significant difference between the virgin treatment and the control injection treatments, which are my secondary controls here. But there is a significant difference between the injection and infusion treatments. So this indicates that it is pre-infusion with DMS, which is causing the microplastics to be ingested at an increased amount. This species here really didn't like the beads at all, but it really enjoyed the fibres and fragments. So to conclude with my shape experiments, the shape of microplastics does influence their uptake in 3 different species. So Calanus ate more fragments, lobsters more beads, and Acacia more fibres. We're not exactly sure what it's down to because further behavioural studies would be needed, but the selectivity of the species, feeding mechanisms, the ease of handling and ingestion, or similarity to prey items are all possibly playing a role in this bioavailability. And then finally, looking at, to conclude with my infochemical experimental results, we show that the pre-infused infochemicals of DMS and DMSP increased the ingestion rate of microplastics for all species, significantly so in the case of DMS for several of the shapes in all species. It's clear that this is the DMSP and DMS are chemoattractants at these concentrations, and therefore it suggests that these species are at an increased risk of consuming microplastics if they mimic the scent of prey. However, we do need further investigation into the detection thresholds, species on certain infochemicals. We currently don't know much about the ability of microplastics to gain infochemical signatures from biofilms. Biofilms are very complex and there's a lot going on, and these experiments are very much a simplification of the visual biofilm effects and also the smell and scent of biofilm. And also there's possibly other different infochemicals out there which may also have an effect, but they may also have a counter effect. There's still a lot more to be done. So I would just like to thank all my wonderful supervisors, colleagues and mentors for all their help so far with my PhD. If you have any questions, let me know. Thank you very much.
That was brilliant. Thank you, Zara.
My pleasure,
so
thank you.
If anyone has any questions, then you can either type them in the meeting chat or you can raise your hand using the little hand button. So while we're waiting for some questions, when you were looking at the colours, did you specifically choose the ones that you'd seen previously on the beach, or did you use a wide variety of?
So I only used one colour, and to be honest with you, I was very much constrained with what I could find out there. So I ended up using a yellow because that's what I could get in all 3 of my shapes. And so the beads I bought, I made the nylon fibres and the fragments I bought, but it is hard to get all your different shapes, plastics being the same colour. You can possibly dye microplastics. Different polymers will take up the dye in different effects. different amounts. But I didn't want to use a dye with different chemicals, which could, of course, then maybe affect their uptake. So that's why I kind of ended up choosing yellow. It wasn't really a choice, it's just kind of what I could end up with, really.
So kind of follow on, if you could get any colours, which ones would you prefer to have used?
So we typically find a lot of blue microplastics, fibres, and a lot of black, browns and clear fragments. So that's probably what, you know, if you were to maybe look at something to do with colour, you'd want to focus on more those kind of colours which are commonly found, but also they do kind of mimic what we kind of find in the natural environment as well, or in, as you can imagine, if you're in the ocean, you're a visual predator, what you may easily, you know, mistake for food.
Okay, so if anyone's typing, then you can raise your hand instead and ask a question. Okay, so Louise has asked, can the organisms you looked at tell the difference between colours?
So that is a good question. Copepods do have a light sensitive kind of eye area, but we don't think it could be able to actually tell the difference between colours. So that's why I wasn't really so worried about it for my experiments. The lobster larvae do have a much more developed nuclear eyes, eye stalks they have, 2 of them, and they probably can tell the difference to a certain level, but the visual kind of effects in zooplankton looking at microplastic hasn't really been looked at. A lot of things have used fish and the colour of microplastics because obviously they're a much more visual predator, but in zooplankton not really much has been done, and in my work I don't think they would have been able to tell a difference, especially because I also ran my experiments in the dark. This is because typically copepods come to the surface and feed at night, so I wanted to mimic those conditions.
Okay, brilliant. So Stephen has said, hi Zara, great research. Maybe I didn't catch it earlier. How did you measure the ingestion rate?
So we use a typical grazing experiment, and we looked at, I had a bottle where I didn't add any copepods to, and I spiked that with plastics. So this was my T0, so I then used this putting through the flow cam. And then after all of my organisms had eaten and I finished my experiment, I had my T6s. And basically you compare your T6s and T0s, and using it's called the Frost's equation, and it's used a lot with zooplankton grazing experiments. And using that equation, we were able to calculate the ingestion rates of plastics. We had to modify it slightly because it's usually used for algal prey, and of course with algae they're going to multiply and grow. Plastics aren't, hopefully. So we just had to take that parameter out, but the same equation can easily apply for that. So that's how I calculated the ingestion rates.
Okay, cool. So we've got another question. They've said, because let's say zooplankton with microplastics will be eaten by a predator and then go through its gut, does that mean that the microplastic will be kind of circulating for a very long time? And is there a solution?
So this is an area of quite a lot of research at the moment. We currently don't really know how long microplastics are retained by zooplankton. So we're kind of wondering, if you have a copepod and they eat plastics, how long does that plastic stay inside the copepod? A lot of factors can affect this, the size of the plastics and also the shape. So logically, slightly bigger, rounder, like beads, microplastics, in theory, should pass through your copepod much faster than fibers or fragments, because they can get kind of caught up in the gut epithelium. But understanding the retention rate is really important, because, as you say, then when these copepod are eaten by predators, this can then end up in the predator, and so on and so on. But until we really understand how long they're retained for, we just don't really know. They could be retained for just a matter of hours or minutes, and then actually the level of threat to other species that would consume the zooplankton is quite low. But we'd have to look at that in many different species. Copepods are one of the most numerous organisms in the world, so I think a lot of work is going on with that. But yeah, until we understand how long they stay inside a copepod for, and crucially the size of the microplastics, so smaller microplastics are probably going to stay longer, and even nano microplastics could have potential effects as well.
Cool. Okay, so that's all the questions. Then just want to say thank you to Zara for coming and presenting at our virtual seminars, and we'll be having another seminar next week, everyone, so I'll see you all next week. And we've got some thank yous on the chat as well for you, Zara. So Louisa said, very cool, thank you, and Stephen